after the BRAF story, I think the most interesting story in the last year has been the ALK story, and it is my pleasure to welcome Alice Shaw. She's an assistant professor of medicine at HMS and at Mass General who works in lung cancer. Alice?
Skip over that. Thank you. So thank you, Nupur. It's a pleasure to be here and to present on behalf of all of the phase 1 Crizotinib investigators, the ALK story. As most of you know, ALK and the targeted therapy Crizotinib have really been catapulted into prime time, culminating last week in Dr. Eunice Kwok's New England Journal of Medicine publication reporting the activity of Crizotinib in ALK patients. So I think now is a good time to think about and review sort of the factors that really contributed to the rapid development of Crizotinib in the clinic. And here are 3 factors that I think were the major contributors, and this will serve as a general outline of my talk today. So first is the molecular target itself, that being ALK, and specifically EML4 ALK. ALK is actually a remarkably good target. It's really not expressed in almost any tissues in most patients. And in addition, ALK rearrangements, which we'll talk about, do confer oncogene addiction. The second factor has to do with our ability to screen rapidly patients for the presence of these ALK rearrangements. And at MGH, John Iafredi in pathology really spearheaded this effort to develop a screening test for ALK, and this has become now the gold standard for diagnosing ALK-positive lung cancer. This clearly facilitated patient selection for the phase 1 trial. And finally, of course, we'll end on the targeted therapy, Crizotinib, also known as PF-234-1066, which is a small molecule tyrosine kinase inhibitor targeting both ALK as well as MET. So because not all of you think about lung cancer on a regular basis, I just included this slide as background. Most of you do know, though, that lung cancer is associated with a high mortality. That's what this slide shows. In addition, what I wanted to emphasize in this slide is that not that much progress has been made over the last several decades. I think this will start to change now that we have these new targeted therapies. I think where we've made the most progress the last 5 years or so in lung cancer is defining and really understanding now that there are molecular subsets of lung cancer. So lung cancer is not all one disease. Lung cancer is in fact made up of different diseases, and in many cases the subsets of lung cancer can be defined by specific oncogenic driver mutations. So shown here is what we know so far about lung cancer, and in particular non-small cell lung cancer. You can see that we know the most about, or we have the most patients who have activating KRAS mutations. We also have quite a few patients with activating EGFR mutations. Shown by the yellow pie, the yellow slice in this pie is the proportion of patients who have ALK rearrangements. It is on the smaller side, roughly 3 to 4% of non-small cell lung cancer patients. But given the large absolute number of patients, this still works out to about 6,000 to 8,000 patients per year in this country alone. So it's a significant number of patients. So back in August of 2007, Dr. Hiro Mano's group first reported the discovery of this new oncogenic fusion kinase in non-small cell lung cancer. And what they did was they actually did a very sort of traditional oncogenic assay to identify transforming genes, and they identified this gene from a patient with non-small cell lung cancer. EML4 ALK refers to this fusion protein that's shown here in the middle, where the amino terminal portion of EML4, which is a microtubule-associated protein, is fused to the entire tyrosine kinase domain of ALK, anaplastic lymphoma kinase. And this creates this constitutively activated fusion kinase EML4 ALK shown in the middle. Now, Dr. Mano went on to show that this fusion kinase has potent oncogenic activity, both in vitro and in vivo. Shown here is one example of his experiments. Here, 3T3 cells were transfected with various expression plasmids. You can see in the red box that the expression plasmid encoding EML4 ALK fusion was able to form foci in these transformation assays, as well as subcutaneous tumors in nude mice. Now, next to the red box is an EML4 ALK fusion that actually has a point mutation at residue 589 that now kills the kinase activity of ALK. And you can see that now you completely abolish the transformation activity of EML4 ALK, showing that the kinase activity of the EML4 ALK fusion is critical for transformation. Now, Dr. Soda, together with Dr. Mano, went on to generate now a transgenic mouse, and this was to address two important questions. The first is the EML4 ALK fusion sufficient to drive tumor formation? And the second question is, are these EML4 ALK-driven tumors now addicted to ALK signaling? So here he generated a transgenic mouse in which EML4 ALK fusion was driven by a lung epithelial-specific promoter, SPC, stands for surfactant protein C. And you can see in panel B that in all of the transgenic animals from the time of birth, there were numerous pulmonary adenocarcinomas. By H&E staining, as shown in panel C, you can see that these were all adenocarcinomas, and shown below is the immunohistochemistry showing expression of ALK in all of these ALK-driven lung cancers. So Dr. Soto went on now to test the activity of a small molecule tyrosine kinase inhibitor of ALK. This is not Crizotinib, this is a different drug. And here he took his transgenic animals, divided them into two cohorts. The cohort on the left, panel A, are animals that are treated with vehicle, and you can see over about a 3.5-week time period that these mice do gradually develop tumors. They're shown in these hatched bars, and they're slowly enlarging over this period of time. And panel B are the animals that were treated with the ALK inhibitor. And you can see over the same period of time that these tumors now dramatically regress. And this was confirmed histologically as shown below in C, where the control animals have these large sort of purple appearing tumors. And in contrast, those animals that were treated with the ALK inhibitor now show much smaller tumors, much less extensive tumor burden. These experiments taken together show you that these ALK-driven tumors really are addicted to ALK signaling and are sensitive to inhibition of ALK. So once Dr. Mano reported his findings, Dr. Iafredi started working very quickly on a way to identify patients who might harbor ALK fusions, ALK rearrangements in tumor samples. And to do this, he actually developed this FISH assay, which has now become the gold standard for screening patients. And briefly, what this FISH assay involves are two probes, a red and a green probe, that flank the highly conserved translocation site within ALK. In the presence of a rearrangement, these two probes are now separated, resulting in a splitting of the red and green signals, as shown by the red arrows in this positive sample. There are other ways to actually identify ALK rearrangements in lung cancers, and here are the two other main ways. We actually did, once John was developing the FISH assay, we did go back and validate it and confirm the presence of ALK rearrangement using these two assays. One is a RT-PCR assay, as shown in A, and the second is an immunohistochemistry assay, shown in B. So right now, we've been primarily using the FISH assay, but one day, certainly the immunohistochemistry may become our diagnostic test of choice. So now with this screening test in hand, we are now actually able to screen our patients that were seen at MGH. Actually, now there have been quite a few case series published from around the world reporting on ALK patients, and so rather than tell you about all of those, I was actually going to share with you our experience at Mass General. So over about a 2-year period from December 2007 to December 2009, we screened almost 500 patients for the presence of ALK. These were screened through our medical oncology clinic, so these were patients who, the majority of whom did have advanced or metastatic disease. The patients were an enriched cohort, and that kind of reflects the referral bias that we have at Mass General. As you can see here, the patients were, actually there were many more females than males that we screened. We tend to see a lot of never and light smokers in our clinic, so you can see that roughly half of our patients were actually never or light smokers, which is much higher than what you would see in the general unselected population. We have a fairly ethnically homogeneous population. Most of our patients are Caucasian. And as I just mentioned, many of the patients that we screened, close to 70% of them already had metastatic disease at the time of screening. So what we found in our collection of patients, is the following. We found that actually among the 500 patients, there were about 9% who had ALK translocations or rearrangements, and there were specific clinical features that were associated with ALK rearrangements. The first one that has been actually shown in a variety of case series from around the world is that these patients do tend to be young, on average 10 to 15 years younger than non-ALK patients. In addition, we found a roughly equal distribution of men and women with ALK-positive lung cancer. And remember, we screened more women than men, so there appears to be a slight male predominance. And then, like the other sort of major subset of lung cancers that we know a lot about, EGFR, we know that we found that these patients, by and large, are almost all never or light smokers. As you can see here, less than 10% of these patients had greater than a 10-pack year smoking history. And finally, something that's somewhat intriguing and that needs follow-up is that we found that many of our patients with ALK-positive lung cancer did have even more advanced disease than the patients that we screened, suggesting that ALK may be associated with an aggressive biology. So this brings us now to the drug that targets ALK, Crizotinib. As I mentioned earlier, this is a small molecule tyrosine kinase inhibitor. It targets both ALK as well as MET. In fact, it was developed early on to target MET-amplified cancers. This selectivity profile of Crizotinib is shown here, and it's meant to remind me to mention that really at the doses that we use clinically, the kinases that are expected to be targeted really are just MET and ALK. So this is the trial design, and I should mention that even before we knew about ALK in lung cancer, this trial was already planned to include 2 phases as shown here, a dose escalation phase, sort of the standard dose escalation phase, followed by a dose expansion phase at the maximum tolerated dose that was meant to allow investigators now to focus in on the activity of Crizotinib in a defined molecularly enriched population of patients. We actually ended up enrolling the first patient in cohort 5, which was at 300 milligrams BID. This is the first patient that was identified with ALK-positive lung cancer at MGH. Now, this phase 1 trial is actually an international multi-center trial with the major sites shown here by the red stars. At MGH, the trial was led by Dr. Eunice Kwok and Dr. Jeffrey Clark. But there were 6 other sites as well outside of Boston, including New York, Chicago, Colorado, UC Irvine, Australia, and Korea. Now, the data I'm about to show you is the most updated data we have on the phase 1 trial. This data is updated as of August 7th, and actually it's a little updated from Eunice's New England Journal publication. Here we've included over 100 patients that are evaluable for safety and response. So this is actually an old slide from ASCO, but this is just, again, meant to remind me to tell you about the clinical and demographic features of the patients who enrolled on the phase 1 trial, and it's pretty much what we have seen in our own patient series, that most of these ALK patients are younger. Most of these patients are never or light smokers, and almost all of these patients do have adenocarcinoma histology. The last thing to note on this slide, actually, and this is an important point, is that most of the patients who went on to this phase 1 trial were heavily pretreated. They almost all had received at least 1 prior treatment, and about half of them had received 2 or more prior treatments. So shown here is the waterfall plot of 105 patients. I'm sorry, the colors, actually there was an Apple to PC conversion mistake, so the colors don't quite match up, but I think most of you can get the idea here that there were a few complete responses. There were many partial responses. The response rate was close to 60%. There are about 30% more patients who qualified as stable disease, just under 30% tumor reduction. And then the last point here to make is that there were some patients, a minority, less than 10%, who actually did have disease progression as their best response, suggesting that in some patients, although we believe that ALK does confer oncogene addiction, some patients somehow lose their oncogene addiction. So this is to summarize and give you some numbers on the response rates that were seen in the phase 1 trial. Again, this is data that was presented at ESMO last month, so this is updated from the paper last week. The response rate was on the order of 56%, with 2% complete responses. The response rate appears to be independent of performance status as well as number of prior treatments. You can see that it holds fairly steady, independent of these various factors. And shown here is the median progression-free survival curves. The median PFS does appear to be on the order of 9 months. And just to get to this point of progression, I mean, the responses are fairly heterogeneous, as are the relapses, and we'll probably come back to that in a minute. But the median does appear to be on the order of 9 months. This is quite a bit more than what you would expect for a patient with metastatic lung cancer who's received prior therapies. Most single-agent chemotherapies in that setting have a median PFS on the order of a few months. So this is some data that we put together and presented at ASCO this year. This is getting at the question, which is very important, of overall survival, and really, does this tyrosine kinase inhibitor also improve overall survival? For those of us who have treated these patients, we certainly believe they do, but we need the numbers to show this. So this is actually patients that were seen at MGH only, so this is a single institution. This is taking all the patients that we've identified who have ALK in their lung cancers, and about 2/3 of those patients have gone on to the phase 1 trial. So this the red curve here represents all of the ALK patients, the majority of whom have gone on to the phase 1 trial. The pink curve here shows just the subset of ALK patients at MGH who never went on to Crizotinib for various reasons. And you can see there's a very clear difference in survival. We're now in the process of putting this data together from all of the phase 1 sites, and we have a preliminary analysis that shows that there will be a significant improvement in overall survival conferred by Crizotinib. So I wanted to just show you 2 examples of the types of responses that we see, and this is kind of similar to what Dr. Flaherty showed as well with his in his melanoma talk. But these responses can be quite rapid and can be quite dramatic. So this is a patient of ours at Mass General who is a 50-year-old woman, never smoker, who had failed 2 prior lines of therapy. At the time she started treatment, her scans are shown here on the left, the pre-treatment scans, and she was symptomatic from her multifocal lung disease. After just 8 weeks of treatment, you can now see her scans on the right, which show a dramatic resolution of her multifocal lung opacities. She's typical in that she reported an improvement in her symptoms within about 1 week of starting on therapy. She actually continues on trial today, a little over 2 years out. This is another patient of mine who I saw at Mass General earlier this year. He was also 50 years old, never smoker, had failed multiple prior lines, and was very, very sick actually at the time we saw him. You can see his scan on the left shows significant lung burden. He actually had widespread metastases as well. And after being on Crizotinib for just 12 weeks, you can see again a very dramatic improvement in his disease burden, and he also had a very significant improvement in his symptoms very early on. This patient also continues on trial now about 7 months out. So, as I mentioned earlier, ALK really is not expressed outside in normal tissues, and I think this explains why the safety profile is so, I would say, relatively spectacular. As you can see here, in terms of the adverse events, most of the adverse events are pretty mild, mostly grade 1, some grade 2. They're primarily GI, nausea, vomiting, diarrhea. There are some other notable side effects. Again, they're still very mild, but they're, I would say, fairly unique to this drug, which include peripheral edema, some visual changes when patients have to adapt from a dark to a light setting. One that is an important adverse event to note is the hepatotoxicity that has been seen with Crizotinib. In about 10% of the patients, there can be LFT abnormalities. They occur at a very typical, stereotypical point in treatment, typically around 4 weeks after starting on Crizotinib, and you can see that there were some grade 3 as well as grade 4 transaminase elevations. Many of these patients who experienced these LFT abnormalities held their dose, and many of them were able to restart at a lower dose. The last AE that I should mention here that's not shown is pneumonitis, and there is the possibility that this drug may rarely cause a drug-induced pneumonitis, but this is still under investigation. So the data from the phase 1 trial has now led to several trials that are ongoing. Actually, again, these are international trials. Profile 1007 is a large randomized phase 3 registration trial. And the key criteria for this trial are that patients have to have ALK rearrangements as demonstrated by FISH. And these patients all have to be in the second-line setting, so they have to have failed 1 prior platinum-based therapy. These patients will be randomized to receive either Crizotinib or a standard single-agent pemetrexed or docetaxel. Now, this trial was not designed as a crossover trial. However, technically, patients who are randomized to the chemotherapy arm will be able to cross over and receive crizotinib. What will happen in this setting is the patients, if they progress on the chemotherapy, they will come off of trial and they will now be eligible for a companion phase 2 trial referred to as Profile 1005. And these patients will now receive crizotinib. So these patients will never be deprived of the opportunity to receive the targeted therapy. Now, Profile 1005 is also meant to capture all of the other ALK patients who are beyond second line. So we see many patients who are now on third line and fifth line and even beyond. And those patients would not be eligible for Profile 1007, but they are eligible for Profile 1005. And future crizotinib trials are shown here. There is a first-line trial that is under development. This trial will hopefully open first quarter of 2011. It's very similar in design as the second-line trial, except these patients are treated up front with either crizotinib or standard combination platinum-based chemotherapy. And also in discussion is the possibility of an adjuvant crizotinib trial to address whether or not an adjuvant tyrosine kinase inhibitor in the setting of ALK-positive lung cancer also can show a survival benefit. So to summarize, patients with ALK-positive lung cancer respond very well to crizotinib, and this is regardless of the number of prior treatments, the gender, age, and performance status. We've seen that to date, the median PFS is on the order of 9 months, and some patients, as I showed you, can have actually very prolonged benefit. Our longest patient is over 2 years at this point. Crizotinib appears to have a very good safety profile, and I should say that our patients actually generally tolerate this medication extremely well. And certainly these trials, the data from the phase 1 support the notion that crizotinib may become a new standard of care for patients with ALK-positive lung cancer. So this is a timeline that actually Eunice and Pfizer and others have put together, showing you, or at least reminding you of sort of the remarkable time frame in which crizotinib has developed over the last couple years. As you can see that actually the phase 1 trial initiated a full year before ALK was even discovered in lung cancer. It was summer of '06. And about a year later was when Dr. Mano reported his findings. And because of the rapid screening that we were able to perform at MGH, we were able to start our very first ALK patient on this trial in December of '07, so just about 4 months after the report of ALK in lung cancer. And certainly, by as I mentioned, the phase 3 trial is now underway, and we enrolled our first patient in the phase 3 trial just earlier this year. So it's pretty remarkable. But so we don't become too smug about everything, I wanted to just end on this slide, which shows you that we do have a bigger challenge now, which really has to do with crizotinib resistance. And these patients, as I mentioned, the response is somewhat heterogeneous. The relapses can also be somewhat heterogeneous. But this is an example of a patient who I showed you earlier who had a dramatic response, has been on crizotinib for over 2 years, and is now beginning to relapse. And you can see her relapsing sort of films here, where you have a small little nodule coming back at about 1 year of crizotinib therapy, and now at 2 years, you can see that this area has clearly grown. So we're working with the Engelman lab to now begin to understand these mechanisms of resistance. For those of you who read the New England Journal last week, you know that one mechanism that was reported by Dr. Mano has to do with a point mutation within the tyrosine kinase domain of ALK, and there probably are other mechanisms as well. So let me just end by acknowledging all of the phase 1 sites who participated in this trial. At Mass General, as I mentioned, Dr. Kwok and Dr. Clark led the phase 1 trial at Mass General and are actually continuing to run the phase 1 trial for other patients, including some of our ALK patients who are not eligible for the phase 2 and phase 3 trials. And also to thank Dr. Iafredi for all of his work. He screened all of the patients that went on to the phase 1 trial. Thank you very much. I can take questions.
Time for a few questions here. And if people are getting their thoughts together, I'll just ask you one. Sure. Alice, obviously the success of this was based on predetermining, ALK status and genotyping these patients. And this is a lesson we've learned from the EGFR story, which took a long time, and we hadn't enriched the patient population. So how robust is this? I know you're doing most of it in central laboratories right now, but moving forward, you're going to need to have a robust FISH test. And I know we can do it at Mass General, but how reproducible is this so that an ALK inhibitor can be used by everybody going forward?
Yeah, so actually, Mace Rothenberg will probably address the question of diagnostics, but I should say that the FISH test can be a little bit finicky, so it's a very valid point. At Mass General, we have a lot of experience with the FISH test. For the phase 3 and phase 2 trials, there is a central lab who is performing the FISH test, and that we've already encountered some issues, potential issues there. So going forward in terms of what the test will be, I would say it's not entirely clear. I showed the immunohistochemistry because I do believe that in the future, immunohistochemistry will probably be technically a more accessible assay for especially local labs to do. So we're waiting for commercially available antibodies that will allow us to have a very robust immunohistochemistry assay. But for now, the current standard is really to use FISH at the central lab.
Alice, there's some unexplained clinical characteristics for both EGFR and ALK, and you mentioned the kind of bizarre female to male sex ratio, which seems inverted. There's also the increased prevalence in Asians, and then there's the whole pathology of multifocal disease with some bronchoalveolar differentiation and so on. Are these all tracked differently between ALK and EGFR, or do you have thoughts about what the mechanisms might be there?
I mean, you're right, there are some interesting similarities and yet differences between these two subsets. I would say they're both never smokers subsets of lung cancer. I think it's pretty clear that these patients, clearly most of them are never smokers, but I would say the difference is we don't understand fully. So the age is a very, I would say it's a pretty marked observation that multiple groups have made that these patients are on the young side, and we have very young patients, even in their late teens, who have ALK rearrangements, and we don't understand that either. So we don't understand the gender, sort of the gender bias, and we don't understand the age distribution. I think it's just too early yet to know all of these relationships.
Alice, thank you very much for a superb presentation. It occurred to me for the members of the audience, you might find of interest in the issue of the oncologist that's at each of your seats, an editorial written by our host, Bruce Chabner. This is not a paid political announcement by Bruce. In fact, it was attributed to a superb spy network at Mass General that the editorial appeared before the New England Journal article actually was published. And I commend it to your attention. If you haven't read it, please do so. And if you haven't talked with Bruce about it, be prepared for a very enlightening and engaging discussion.
Sorry, hi, Alice. The question is, you made the statement that this disease is predominantly a non-smoker disease, but in the numbers that you showed, it seemed like at least 25% of the patients have either had light smoking histories or heavy smoking histories. So does the heterogeneity of primary response track with a previous history of smoking at all?
You know, there haven't been enough significant smokers to really track that, at least I would say in my experience at MGH. But I should say what I called smokers is greater than 10 pack years. Many of those patients were still less than sort of the average smoker you might see, so they might have been 12 pack years or 15 pack years, relatively on the light side. And then also the so-called light smokers were people who might have only smoked for 2 years in college, and they're called light smokers. So if we were able to go back, as we accumulate more patients, we'll go back and look probably by sort of pack.
Exception in prostate where there's like a 2% frequency of a translocation to BRAF that was discovered and published in Nature Genetics by Rulchinayan. Those are sort of the things that have emerged so far.
Thanks. What have you learned about the MET side of the story? As you know, this was initially thought to be working in the MET amplified, and what have we learned on the MET side in lung cancer in these trials?
So, I mean, I would say that the initial reports of MET amplification in lung cancer, I think in reality the estimates were probably too high initially because we see very, very few cases of MET amplified lung cancers. There are other MET amplified tumors though, and some of these patients have enrolled on the phase 1 trial actually. And I guess it's anecdotal still, so I can't really comment, but it's not clear yet to me how well it works for MET amplified cancers, and that may be a function of the drug, or it could be a function of the fact that perhaps these tumors aren't as addicted to MET signaling.
Given the length of time it took to learn about the incidence of EGFR mutation in African Americans, I'm wondering what efforts have been made to get tumor from African American patients for the ALK trials?
So, I mean, I know the sponsor is actually looking worldwide for collaborators to address this very question about the distribution of patients by ethnicity. What are the frequencies like? We already know that in Asia there is a higher incidence of ALK. I'm not sure, I don't know the details. We can actually ask Dr. Rothenberg about this in terms of looking, for example, either in the US or in other, for example, African countries at the incidence in African Americans.
So one more question. When the response rate is this high as 56%, got to ask the question about those handful of patients who are showing the progressive disease as the best response. What do we know about them? Is that a diagnostic error, or is it a different biology, a different translocation?
It's a very good question. We don't know. I can only comment on the patients that we've had at MGH who have had sort of progressive disease as their best response, and we had 2 of them. And we did go back with Dr. Iafredi and review those to make sure that they definitely were ALK positive, and they were confirmed ALK positive by FISH. And by, in at least one case, by immunohistochemistry as well. So we believe that at least for 2 of those cases that I showed you, they were truly ALK positive. But as I mentioned, we don't understand yet why a small minority of tumors do seem to lose their addiction. We don't know why.